cholesterol metabolism. The topics included in this lecture are cholesterol metabolism, regulation, as well as hypercholesterolemia. Talking about the regulation of cholesterol synthesis, regulation of the levels of cholesterol in the circulation as well as in the tissues occurs at different levels. The regulation of synthesis and uptake. As the cellular content of cholesterol increases, it exerts a sort of feedback regulation where the elevated cellular cholesterol itself inhibits the sense of the regulatory or key regulatory enzyme, hydroxymethylglutarate-CoA reductase. Also, there is reduction of low-dense lipoprotein receptor synthesis, and these two processes contribute to subsequent reduction of cholesterol uptake. A third effect is activation of an enzyme, acetylcholesterol which is concerned with cholesterol storage, whereas cholesterol is acetylated or esterified and stored in the form of cholesterol ester. Another level of regulation is exerted as a transcription level of hydroxymethylglutarate-CoA reductase gene. And as mentioned earlier, cellular cholesterol is a major effector of this process. The transcription of hydroxymethylglutarate-CoA reductase gene is regulated by a protein SREP or steroid regulatory element binding protein, which is synthesized and activated in the endoplasmic reticulum and Golgi, then enters the nucleus where it binds to steroid response element on the DNA or the gene promoter, where transcription starts. And as shown in the figure, elevated cellular cholesterol negatively affects the SREP, where SREP is bound to another regulatory protein, which is SCAP or SREP cleavage activating protein, leading to cleavage of SREP with the reduction of hydroxymethylglutarate-CoA gene expression, or the HMG-CoA reductase gene expression. Back to the regulation of cholesterol synthesis, we have the hormonal regulation. After meals, insulin induces activation of hydroxymethylglutarate-CoA reductase by activating a protein phosphatase that dephosphorylates the hydroxymethylglutarate-CoA reductase with subsequent activation and increased cholesterol synthesis. So, as we see, feeding promotes cholesterol synthesis by providing the substrates for cholesterol synthesis, which are acetyl coenzyme A and the reduced NAT phosphate, and activation of the regulatory enzyme hydroxymethylglutarate-CoA reductase. During fasting and the periods of food deprivation, we have glucagon, the hormone that activates adenyl cyclase with increasing levels of the cyclic AMP and subsequent activation of a protein kinase that phosphorylates the regulatory enzyme hydroxymethyl-butyrate-CoA reductase and inhibits its activity. Subsequently, cholesterol synthesis is reduced. This process is AMP dependent, so when ATP is reduced, there will be no need for cholesterol synthesis. So, as we see that fasting is a good cholesterol lowering strategy where the hydroxymethyl-butyrate-CoA reductase activity is reduced. So, as you see, the covalent modification of the enzyme is key for regulating cholesterol synthesis. The hydroxymethyl-butyrate-CoA reductase is covalently modified by phosphorylation-dephosphorylation reaction. The active form is a dephosphorylated form. This is achieved by a protein phosphatase that is activated by insulin, whereas it is inactivated by phosphorylation by a protein kinase, which is activated by the hormone glucagon during fasting and starvation. And hence, this explains the role of fasting in down-regulating enzyme activity and cholesterol level reduction. To summarize the previous regulatory mechanisms for the activity of hydroxymethyl-butyrate-CoA reductase, we have short-term regulation by phosphorylation, dephosphorylation, and long-term regulation by proteolysis of the enzyme itself and cholesterol-stimulated degradation of the enzyme. Also, gene expression and regulation by the sterol sensing domain and the strep protein. You can find other relevant information about this lecture and other lectures at this site.